The cell is a machine. It's a factory with many modules, which are sub-machines that do certain functions. Understanding how these machines work independently from each other, understanding how the DNA in the nucleus is being copied, is being edited, how mistakes are being corrected, all this is the subject of current interest. And then, of course, understanding how the machines are actually coupled together, that it's not kind of a random orchestra, each playing its own part. Well, the thing that has changed is that for the last 10 years we have been able to study these machines one by one. People have developed tools that allow the manipulation of single molecules. We can look at one molecule at a time, learn exactly how that molecule works, compare it to a similar motor from another organism or from the same organism, and try to learn if there are common principles. So the first tool was known as the scanning tunneling microscope or the family atomic force microscope, etc. But following that, people started to develop optical tweezers and now magnetic tweezers, and there are all kinds of techniques that allow people to actually pull on single DNA molecules, pull on proteins, and also look at protein-protein interaction. And there are machines known as topoisomerasers, and they perform certain function on the DNA. So what they do basically is that when the two molecules are entangled like that, they open one molecule, and then the other can pass through, and then they reseal the other molecule. And this way, if molecules are stuck to each other, they can be unstuck. If they malfunction, the DNA cannot be disentangled, cannot be separated, and when the machines are pulling on the chromosome, they basically tear them apart. And there are other machines again that transport these subcomponents to the place where they are used, like trucks are transporting cars that are made in a certain given place to a town where they are being sold. So the machines that do that are known as kinesins. Basically, its role is to carry, to transport cargo from one place to another. Some of them will transport nerve impulses along the neurons. Some of them will transport all the proteins that are being required at the end of the axons. So the track that brings things from the cell body to the axon is kinesin, and the tracks that bring things from the axon to the cell body is known as dynein. And it turns out that they move like a human being. They have two legs, and one leg goes from being in the back to being in the front, and then the back legs goes back in the front. The goal will be to understand how they work, what is the principle of organization of these things, to be able to design ourselves chemical motor, artificial motors that could also be used in nanotechnology to make smart materials, smart polymers, smart components, electrical components, to couple the biological world with microelectronics or with chemistry, but we are still very far from that.